where she entered the lab in, I think, May 2017. That's probably the time when people decide where to join. And she immediately, within a few weeks, ran a screen. And that screen happened somewhere in summer 2017. Immediately got something out of that screen, ran with it, wrote up a paper, and within a year had a bioRxiv paper out. And that then finally came out in Bio in January 2019. And there's another remarkable thing about this paper, because another thing in grad school that you don't usually get while you're still in it is the validation that other people pick up your work. And Shannon got that immediately, and she's going to talk about it as well. So I'll just briefly mention that at the very same day that this paper came out, another group published basically exactly the same thing in a different organism. And that's a really nice validation, and we weren't aware of them, they weren't aware of us, but it just happened to coincide like that. So Shannon is really a prolific author of manuscripts. And since she's only going to talk about one of them in her talk, I'm going to point out the other ones. She will graduate with not one, not two, not three, not four, but 5 papers. And the last one is in bioRxiv. It's currently under review in PLOS Genetics, and of course it'll be accepted there because the science is flawless. It's also an interesting one because this was really a truly independent Shannon project, where Shannon found this really interesting phenotype and just ran with it, even though the subject area does not really touch upon the core identity of the lab. But it's just, I mean, that's the best thing that can happen to you in grad school, right? You find something that you're interested in and you just focus on that and run with it. That said, since it is not the core of my identity, of course I had to resort to some passive aggressive remarks. And naturally, I did that on Twitter. And the background here is that Shannon is also our tweeter-in-chief. The reason is that I'm old and I don't understand technology. So Shannon basically handles our Twitter account. And 95% of the tweets that you see from my lab are actually sent by Shannon. That said, I do, of course, have the account information. So when I presented her paper to the world, I wrote, "Going where the data drag us." And I sent that out and chuckled a little bit thinking, I wonder if Shannon even notices this little slide. And notice she did, because within the hour, I was sternly rebuked by going where the data leads us. Okay, so basically I feel dragged, Shannon feels led, but we both, I think, ended up in a place where we appreciate the outcome. So that's ideal. Here's another measure of Shannon's productivity. So I went through her supplemental data and the strain lists and counted the number of strains that she has produced in her time here. For her first paper, it was something like 3 or 4, then 6, then over 10. And for the last paper, it was actually over 100. You can immediately see that this is an exponential function, right? So you can crudely fit a Lin log curve in here. So this is the log scale here, then extrapolate a little bit. And so according to my calculation, by her 7th paper, Shannon will have made 1,000 strains just for that paper. This is not accumulative, of course. And given her track record in my lab, I'm pretty sure that number 6 and 7 are still actually going to happen in the 2 or 3 months that she is going to stay with us. Shannon is also a purveyor of undergrad excellence. So she has supervised countless undergrad researchers in the lab. Many of them ended up as co-authors on papers, and they've really gotten a really nice education out of this from Shannon. Here's a picture of a recent lab retreat and 2 of the most recent undergrads that ended up on Shannon's papers. Shannon is a gifted communicator of science, so she has really internalized the need for scientists to communicate their results to the world so that a lay audience can also understand that. And if that means wearing chainmail because that happens to look like the bacterial cell wall, she's absolutely going to do that. And I think it really helps. Well, it's either that, or I don't know, actually, I didn't see this talk. Maybe she's about to present something so controversial that she was fearing actual violent opposition, but that's her communication skills. So I won't spend much more time on keeping Shannon from presenting her work, but Shannon has come a long way from a toddler uselessly idling away on a swing to whatever this is. And has really morphed into a fantastic scientist. So Shannon is a deep and independent thinker. It's a delight to sit here in my office with her and discuss models and hypotheses and how to proceed from various project angles. She's also a prolific and expert writer. So, and this is something that's often not articulated as the primary goal of graduate school, but I think it should be. And Shannon has really internalized that and really improved her writing skills. Actually, the last paper that's in bioRxiv. I remember, I think I spent more time writing things like, wow, that's such a beautiful sentence, or hey, that's a great paragraph, than actually editing it. So Shannon really knows how to write a paper. And part of that is actually her main trade is a literature archaeologist. So when Shannon writes a paper, she takes a deep dive into the literature. And she will end up in the most obscure and darkest corners of some supplemental data in some obscure paper from way back when, and find something that supports her argument and supports her hypothesis. She is, of course, also a highly skilled experimentalist and an outstanding lab citizen. So Shannon, it's been an absolute delight to work with you over the last 4 or 5 years. You will be sorely missed, but it's time for you to move on and do even greater things, and I'm sure you will. So with that, I yield the floor, and you're up.
Toby, thank you for that wonderful introduction. I'm going to go ahead and share my screen. Okay, so I want to just extend a really big thank you to everyone that's joining here today. I have a lot of friends and family in the audience, past mentors, and it's just really comes full circle for me to see you all here. So that is one upside to all of this, is having a Zoom defense means it's more accessible to the people who I'd love to see this. I'll be sharing you with my work today on Vibrio cholerae. This is a bacterium that causes the diarrheal disease cholera. And this strain intriguingly harbors some extra genomic islands that weren't present in past cholera pandemics. It's long been suspected that these islands give V. cholerae a competitive edge over these strains of the past, but truthfully, we know very little about their contents. So my goal for today is to expose some of that island's secrets. Overall, Vibrio cholerae's success as a pathogen is owed to the fact that it can adapt to 2 very distinct lifestyles. It lurks in estuaries and brackish waters, and it persists in these environments by attaching to biotic surfaces, mostly the chitinous exoskeletons of crustaceans, macroinvertebrates, or shellfish. So cholera can be ingested into the host either by drinking contaminated water or eating undercooked seafood. Once inside the host, V. cholerae migrates to the gut and uses pili to attach to the intestinal epithelium. There, it secretes the cholera toxin that causes severe diarrhea, and this rapidly dehydrates the patient with potentially lethal consequences. This diarrhea also sheds bacteria back into a watery environment at very high numbers. And so cholera outbreaks tend to occur when wastewater contaminates the supply of drinking water. To give you a historic example of this, we can travel back in time and in place to 19th century London. The city's population was rapidly expanding, but the sanitation infrastructure was not keeping up. So what this meant is that cholera outbreaks were common and they were deadly. This cartoon from 1866 shows Death's Angel carrying a suitcase of cholera, and the caption reads, "A warning to landlords. You'll have to come down with your rents. I intend to occupy the premises myself." And occupy it did. There was a particularly deadly outbreak in London's Soho district, and in response, a physician named John Snow went door to door, interviewing residents to determine who was sick and where they got their drinking water from. He made a map of all of this data and was able to trace the source of the infection to a singular water pump on Broad Street. And as it turns out, this well was contaminated by a nearby cesspit. Snow alerted the city officials, and when they failed to take action, he ripped the handle off the pump to stop residents from using it, and the outbreak subsided. Now, the story is widely told because Snow efforts were a founding example of epidemiology, which is how we track disease. And prior to COVID, I had the opportunity to travel to London with another student in our program, the Van Way Adams. And while we were there, we visited the former Broad Street pump, where there's a statue commemorating Snow's efforts and work. Like many others during this pandemic, I have a newfound appreciation for things like contact tracing and daily checks. And so when it's safe to travel again, I recommend visiting this quirky point of interest in London to pay homage. That being said, I need to emphasize that cholera is not an issue of the past. So cholera is endemic or regularly found in over 50 countries, causing millions of illnesses and tens of thousands of deaths each year. Cholera outbreaks tend to closely follow humanitarian crises. In the last decade, some notable examples include in Haiti following the 2010 earthquake, some scattered outbreaks in West and Central Africa, and in Yemen. So the Yemen outbreak began around the same time that I was starting graduate school. And in the time that it takes to complete a PhD, there have been more than 2.5 million cases and several thousand deaths. But something that might surprise you is that the cholera agent that we globally face today is not the same agent that John Snow tracked down in the mid-1800s. Over the past 200 years, there have been 7 recorded cholera pandemics. The first 6 were caused by the classical strain shown on the timeline in blue. But following a brief pandemic-free period, there was a new, more dominant player that emerged, and it's called L-Tor, and it's shown on the timeline in green. And L-Tor is responsible for this ongoing 7th wave. So what do these two agents have in common? They're both from the same O1 serogroup, and they also encode similar virulence factors. So both of them have that cholera toxin that causes diarrhea, as well as the attachment pilus that allows for intestinal colonization. But how are they different? So L-Tor is considered a different biotype. It's genetically very similar, but it can be distinguished based on a number of biochemical tests. About 13% of its genome is differentially expressed. And so having these gene products present in different amounts can actually have very important consequences on bacterial fitness. L-Tor is resistant to polymyxin B. And the aspect that I'll focus on today is that L-Tor has acquired 2 new genomic islands. These islands are named VSP1 and 2 for Vibrio 7th pandemic, and each of them bear the hallmarks of horizontal acquisition. They have an unusual GC content compared to the rest of the genome, and they contain mobile elements like integrases or transposases that are shown in blue. For decades, it's been suspected that these islands helped L-Tor to globally outcompete the classical biotype, but we admittedly still know very little about their contents. So many of the encoded proteins remain hypothetical. Those are indicated in gray, and the uncharacterized proteins are shown in yellow. So let me start with what we do know. On the first island, VSP1, there's this gene product called DNCV, which secretes a signaling molecule that decreases motility. In vivo, this is important for colonization, possibly because it allows cells to slow down and better attach to the intestinal walls. More recently, in a bioinformatic analysis, it was shown that DNCV is frequently associated with phage defense systems. Science moves very quickly. Just this week in a preprint, a neighboring gene was shown to corrupt cellular nucleotide concentrations, and that offered protection against phages. So VSP1 seems to offer L-Tor some kind of benefit when it comes to host colonization or avoiding predation by phage. But in very sharp contrast to this, the VSP2 island is 26 kilobases of pure mystery. We do not know if or even how these genes would contribute to pathogenicity or environmental persistence. So why hasn't anyone figured it out yet? Well, one of the challenges of studying this island is that most of its contents are not transcribed under standard laboratory conditions. And without that activation, it can be difficult to decipher the function of these genes. So this left many in the field to say, all right then, keep your secrets. But I think this is a really compelling mystery. And so what I wanted to know is what signal from cholera's native environment induces expression of this mysterious island. So as a preview to the 2 stories I'll tell you today, I'm going to explain how I discovered a signal that upregulates a portion of this island and what we've learned so far about the function of those encoded genes. And I think that there is an immense value in telling you the story how it actually happened. There's this expectation that research will be linear, that you'll ask a question, you'll do experiments, and you'll get an answer. But my journey through graduate school, as Toby alluded, was quite different. And so I started off studying an entirely different aspect of E. colaris biology. And without ever intending to, I answered a question about this island that I wouldn't have otherwise known how to ask. So this kind of path requires a little bit of luck, a lot of curiosity, and ultimately just a willingness to follow the science wherever it leads, not drags. And above all else, this path requires a very supportive advisor. So I'm grateful to Toby for encouraging me to follow the data, even when it really challenged and stretched the bounds of our research identities. So now that I've outlined for you the question that my work answers, I want to show you where I actually began. And that was with the bacterial cell wall. So one of the things that I admire about bacteria is that they are experts at dealing with stress. And one of their best defenses is the cell envelope. It's a physical barrier that can shield them from harsh and fluctuating environmental conditions. This envelope in Vibrio and other gram-negative bacteria consists of an inner and an outer membrane, and sandwiched in between is a very thin layer of cell wall. The cell wall is important because it provides rigid yet flexible support, and it gives a bacterium a defined shape, in this case a rod or a comma-shaped rod, and the ability to withstand strong turgor pressures. As Toby mentioned in a prior talk, I compared the cell wall to a suit of medieval chainmail, and I really committed to the analogy. So much like the cell wall, chainmail has a shape to it, but it also allows for movement. And in terms of composition, the cell wall is also quite similar. It's composed of these repeating peptidoglycan units, which are a sugar disaccharide with a peptide stem. These units are linked together into long strands via transglycosylation, and adjacent strands are cross-linked via transpeptidation. And that's what allows peptidoglycan to form this kind of 3D stacculus or meshwork. The cell wall is a great source of strength and protection against stress, but it is also its Achilles heel, because many of our antibiotics work by inhibiting cell wall synthesis. So in this movie, you'll see that E. coli, when it is treated with penicillin and blocks cell wall synthesis, the cells rapidly lyse. And one of the reasons in the lab that we study the V. cholerae envelope is that it can actually tolerate these drugs. Rather than lysing, the cells completely lose their cell wall and they exist as these spheres. And understanding the mechanics of this superior cell envelope is important for understanding why antibiotic treatments sometimes fail. Now, all of the depictions that I've given you thus far of the cell wall are quite static, but in reality, the cell wall is a very dynamic structure because it has to be. The cell wall needs to expand and remodel in order to accommodate a growing and dividing cell. And this remodeling is achieved by both cell wall synthesis and degradation. So in order to expand, links in this matrix must be cut apart, creating space for the insertion of new pieces or vice versa. And while we know quite a bit about cell wall synthesis, we know comparatively less about cell wall degradation processes. This breaking of peptidoglycan is carried out by numerous enzyme classes. These include amidases and carboxypeptidases, which modify or remove the peptide stem, respectively, lytic transglycosylases, which cleave the glycan backbone, and endopeptidases, which are my personal favorite, cut the crosslinks that hold adjacent strands together. Now, endopeptidases are required for normal growth, but due to their destructive potential, they must be tightly controlled to achieve the morphology that you see here. This is kind of like a Goldilocks situation. If you have too much endopeptidase activity shown here, the cell wall gets chewed up and you lose that defined shape. But on the other hand, if you deplete endopeptidases away, the cell restricts the regrowing cell mass and you end up with what looks like 10 pounds of sausage in a 5-pound casing. So these two images came from 2 other cell wall projects that I was involved with. But to make this regulation even more complicated, these cell wall degrading enzymes are highly redundant in the bacterial genome. V. cholerae, for example, has 6 endopeptidases. And that prompts the question, why are there so many? Why not just have one good one? Well, why do you own more than one pair of shoes? Perhaps these extra shoes or these extra endopeptidases are specialized for a specific set of conditions. Some might be more general purpose, while others are reserved for good and bad weather. So we hypothesize that these extra endopeptidases are environmental specialists that might enable V. cholerae to adapt to disturbances like changes in osmolarity, pH, temperature, or more broadly, stresses that are associated with the host environment. And so with this idea in mind, I would like to introduce you to 3 of cholera's nearly identical endopeptidases. And these are named Chi-A, B, and C. They each have a lyse-M domain, which binds to peptidoglycan, as well as an M23 metallopeptidase domain. This is what actually cleaves the crosslinks that hold those adjacent strands together. And I'll mention here that Chi-B is located on this VSB2 island, meaning that it's present in L-Tor, but not in other V. cholerae biotypes. And this will become important later on. So in understanding why cholera has 3 endopeptidases, I first looked at whether these genes are even expressed. To easily measure that, I created a transcriptional reporter where I fused the promoter of each gene with LAC-Z. So if the promoter is on, LAC-Z activity will give the colonies a blue color, and it'll look like this one on the left. And if the promoter is off, they will stay white. So on a standard LB agar, you can see that Chi-A and C are transcribed, but Chi-B, much like other VSB2 genes, appears to be silent. But why would a bacteria bother encoding a gene if it doesn't express it? Well, perhaps Chi-B is one of these environmental specialists, and maybe it is upregulated in response to a stress under which it is specifically adapted to function. So we can think of Chi-B like the snow boot. It's kept in storage until winter weather rolls in, and then you wear it to keep your feet dry and warm in the snow. So how do we identify the stress signal, and how do we identify the mechanism of transcriptional control? To find out, I ran a genetic screen that used that Chi-B transcriptional reporter, and this was one of my very first experiments that I did in the lab. So I took that reporter strain, which is normally off, and I mutagenized it with Mariner transposons. These will insert randomly throughout the genome, and if it lands in an open reading frame, it will essentially create a gene knockout. And so the goal of the screen was to identify an insertion that induced the Shy-B promoter. And that was as simple as plating the mutant library and looking for colonies that turned blue. I picked all the blue ones and mapped the site of transposon insertion using a technique called arbitrary PCR. So here's what that actually looked like. This is a large agar plate, about 12 inches by 12 inches, and it has about tens of thousands of colonies on it. And you can see that most of them are white, but in total, there were about 24 blue colonies. Some were dark, others were lighter blue. And I expected that these transposon insertions would map to some kind of transcriptional regulator, perhaps something to do with cell envelope stress. But instead, I got a lot of things that had to do with zinc. So all of these dark blue colonies map to ZR. ZR is a transcriptional repressor that governs zinc homeostasis. And these lighter colonies mapped to ZNU, which is a zinc import system. And so this was not at all what I expected. And it was really at this point where I felt like my dissertation were kind of pivoted. I started thinking less about cell wall and more about zinc homeostasis. So I want to provide you a bit of background about why bacteria need zinc in the environments where zinc is in limited supply. And then want to explore all of the many adaptations that bacteria have for responding to this zinc starvation stress. So zinc is an essential micronutrient, and it's required as a structural ligand or cofactor in about 5% of the bacterial proteome. And so these proteins are responsible for carrying out a number of essential processes. But zinc is often in limited supply. So for example, vertebrates, which also use zinc, are very good at sequestering metals in order to restrict bacterial growth. This is an innate defense mechanism known as nutritional immunity, and it creates an intense competition for these metal cations within the microbiome. But bacteria also encounter zinc deficiency when they grow in biofilms. This dense layering of cells has less surface access to micronutrients. But to cope with this deficiency, bacteria express a set of zinc starvation genes. This regulon of genes is expressed only when zinc is low, and its components tend to come in two flavors. They encode things like transporters that help import more zinc, or they encode alternative proteins that are non-zinc binding. And these will carry out those important processes in the absence of zinc. If bacteria then enter an environment that is zinc rich, expressing these genes is a waste of energy and potentially harmful. So these are turned off by the transcriptional repressor ZR. And you can think of ZR as the energy conscious member of your household or lab that goes around flipping off switches at the end of the day. ZR is able to bind and form a complex with these abundant zinc cations. And in this bound conformation, it has high affinity for specific DNA sequences. When it binds, it represses transcription of those downstream genes. Now, because ZR disruption and also disruption of these zinc uptake systems led to Shy-B expression, our screen seems to imply that Shy-B is a novel member of this starvation response. In order to test that, I put that Shy-B reporter on some agar that's made up with minimal medium, which is naturally low in zinc. And you can see that the colonies are blue. But if I then supplement those same plates with zinc, you can see that the promoter gets turned off and the colonies are white. So zinc is the environmental input that's being sensed. If I do the same thing in a ZR mutant, you can say that the reporter is constitutively on regardless of zinc availability. That's because this strain does not have ZR there to physically repress the promoter. So zinc is the signal and ZR is what's actually carrying out the repression. To show that ZR exerts a direct effect on the promoter, I looked for a ZR binding site. And that consensus sequence is shown here, where the height of the letter corresponds to the degree of nucleotide conservation at that site. You can see that the Shy-B promoter sequence is a perfect match, but I also validated this interaction through an in vitro binding assay. So the Shy-B endopeptidase is indeed a novel zinc starvation gene. It's repressed by ZR when zinc availability is high. So now that we've identified the stress that induces Shy-B, I wanted to know, is this enzyme actually specialized to function during zinc starvation? To test this, I examined the expression and utility of individual endopeptidases under both high and low zinc conditions. I did this by deleting all but one endopeptidase. So to examine the effect of Shy-A, for example, I deleted Shy-B and C, and so on. Expression was determined via Western blots of tagged proteins, and growth was monitored by changes in optical density of a culture over time. In a high zinc medium, only Shy-A and C are expressed, and either are sufficient to support growth. But as I showed you before, Shy-B transcription is repressed by ZIR, and so it's no help. In a low zinc medium, all three of these endopeptidases are detected at the protein level, and Shy-B alone can support growth. But what was interesting here was that Shy-C could not. So what our data suggests thus far is that Shy-B, when expressed, can substitute for Shy-C. And that allows V. cholerae to retain some amount of enzymatic redundancy. But why does low zinc availability inhibit endopeptidases? Shy-A and C were previously shown to require zinc as a cofactor to cleave those peptide crosslinks. And this means that their activity is inhibited by zinc chelation. But what about Shy-B? So we tested Shy-B's activity in an in vitro assay with the help of two collaborating groups. So Mavanway in the Chappie lab helped us to purify each endopeptidase, and Laura Alvarez and Felipe Cava in Sweden were able to measure the activity against purified cell wall substrate. So here's what that data looked like. The y-axis here is showing relative endopeptidase activity, and Laura ran these reactions with an increasing concentration of EDTA. This is a general chelator of divalent cations like zinc. So you can see that as we increase the amount of chelation, Shy-A and C are inhibited, confirming that their activity is indeed metal dependent. I want to note here that Shy-C seems to be slightly more sensitive, and our growth data suggests that this difference in sensitivity might be biologically relevant. But what was really striking was Shy-B. Shy-B is highly resistant to chelation, even up to 20 millimolars of EDTA. And that's a concentration that far exceeds the MIC for V. cholerae growth. And this suggests one of two things, that either Shy-B is holding on very tightly to its metal cofactors, zinc or otherwise, or it has evolved to function independently of a bound metal ion. So regardless, it suggests that Shy-B is specialized to function in this low metal environment. And this was pretty novel. It was, at the time, the first report of a cell wall hydrolase that was shown to be part of the zinc starvation response. And its inclusion in this regulon makes sense because unlike other endopeptidases, it is highly resistant to metal chelators. And we published this work in MBio in 2019. And in our final figure of the paper, we suggested that these zinc specialists might be present in other bacteria. I had been searching for ZIR binding sites adjacent to endopeptidases, and I had found a Shy-B homologue that is right next to the ZIR-regulated zinc import system. This motif is present in many gram-negative pathogens listed here, and I dug through published transcriptomic data sets and found that E. coli and Yersinia pestis also seem to upregulate this endopeptidase in response to zinc stress. And so my hope was that this figure would set the stage for other bacteriologists to pay attention to this motif. And in 2020, a group did just that. In the plant pathogen Erwinia amylivora, they showed that this endopeptidase is ZIR-regulated and that it's important for virulence. But sometimes the field moves faster than you expect. So on the very same day that we published our story in MBio, the SCAR lab reported a similar finding in another bacteria, A. bimanii. And on that day, the PI from that lab posted this tweet that I think sums up the sentiment nicely. He said, one day, 2 different papers, 2 different journals, 2 different labs, 2 different organisms, remarkably similar results. And I agreed with this. It's really satisfying to know that the work that you've poured your heart into is relevant beyond just your bacteria of choice. So this was a wonderful tweet to see on a day that I was already excited about. And we were really excited about this new link between cell wall and zinc homeostasis. But what we did not fully appreciate at the time is that we were starting to unravel a decades-old mystery. We had identified a cue, zinc starvation, that triggered expression of the VSP2 gene. Now, knowing that Shy-B is regulated by zinc, a reasonable question would be, does zinc deficiency trigger expression of other genes on this island? But that's not the question that I asked, but I still managed to get there through an indirect route. And this time it was by simply following up on a curious observation. So the second story, like any good microbiology endeavor, starts with an overnight culture. So I had grown a wild type and a ZIR mutant overnight in minimal medium. And when I pulled them out of the shaking incubator the next morning, I saw a very normal wild type culture, but the ZIR mutant looked pretty odd. It had a clear supernatant and the cells were all accumulated at the bottom of the tube. This aggregate was quite loose, and you can see in this video that I can easily disturb the pellet. So as a side quest, I just wanted to know why this mutant aggregated. If you recall that in the absence of ZIR, zinc starvation genes are constitutively transcribed, but I went through and checked every single one, and none of them were required for aggregation. So I thought maybe there are even more genes that we have yet to find. So I did 2 things here. I ran an RNA-seq analysis to define the full ZIR regulon, And in parallel, we ran a screen to identify any and all genes that are required for this aggregation behavior. So in that screen, we took the ZR mutant and we mutagenized it with the same transposons that I told you about earlier. We then grew that mutant library in minimal medium. And while most of the mutants still aggregated at the bottom, we were able to sample the cells that were still swimming towards the top in the center of the tube. We repeatedly subcultured these until there was no visible pellet, and then isolated single colonies and mapped the site of transposon insertion. So together with my undergrad mentee, Bree Johnson, we mapped about 50 of these transposons, and the majority of them landed in motility genes. The most saturated hits were within flagellar components and regulators, and those are shown in the pie chart in yellow. And we also had numerous insertions within chemotaxis genes that are indicated in purple. Chemotaxis is a system that allows a bacteria to swim directionally along a chemical gradient. So to validate these hits, we needed a way to quantify aggregation. So we measured the optical density of the supernatant before and after vortexing and took the ratio. And a ratio closer to 1 means that the culture is homogeneous, like the wild type shown here. And a ratio closer to 0 indicates aggregation, and here's ZR. So we made clean deletions of each gene in a ZR mutant background and checked to see whether they still aggregated. This was done together with another mentee, Camille Ledoux. You can see that by deleting either flagellar motility, rotation, or chemotaxis, delta ZR is no longer able to aggregate. But importantly, we're able to restore these aggregates by complementing each of these genes back in trans. So we can begin to assemble a model for what's going on here. We learned that there is a requirement for a functional flagellum, and that means that V. cauleri is actively swimming to the bottom of the culture tube. And more specifically, it's chemotaxing in response to some kind of chemical incentive. I was kind of surprised by this result. I thought this phenotype would be driven by attachment rather than swimming. And so as a sanity check, I went through and made targeted deletions to quorum sensing, attachment pili, and biofilm formation genes. And each of those were dispensable. So this really reinforced the conclusion that what we are seeing is indeed a motility-dependent process. But none of these hits appear to be ZR regulated. So that kind of left us scratching our heads with what went wrong with the screen. Well, in the words of bacterial geneticist Tom Selhavi, you get what you screen for, but you don't know what you're screening for until you get it. What he means by that is that sometimes your screen is a strong selector for something that you did not anticipate. And that's exactly what happened here. We thought that we were screening for aggregation factors that are uniquely expressed in the ZR mutant. But our bacteria said, no, we won't give you that. It turns out that we had really just designed an elegant screen for non-modal mutants. And as Bree can attest, there were so many motility genes to saturate that it felt like we were missing something important. So we took what we learned from that first screen and decided to work smarter and not harder. We added a step where we pre-filtered the library to select for mutants that could still swim. We did this by inoculating our transposon library into soft agar. The non-modal mutants will stay at the center inoculation site, and the modal mutants will migrate outwards. We pluck cells from these outer regions and then enrich for non-aggregators just like before. So this worked. By excluding those non-modal mutants, this revealed a very strong cluster of hits. And wouldn't you know, these transposons map to 2 neighboring genes on the VSP2 island, VCO512 and VCO513. If we mutate either of these genes, the pellet doesn't form. So I was absolutely giddy over this result because I realized that this phenotype could be leveraged to tell us something very interesting about the function of VSP2. So to summarize the pieces from these 2 screens, we know that aggregation requires a functional flagellum and this chemotaxis signaling system. And we know that 2 VSP2 genes are involved. These encode a transcriptional activator, which I've named VER-A, and a chemotaxis receptor, which I've named AIR-B. But since this phenotype is specific to a ZR mutant, I first wanted to know, are any of these genes ZR regulated? To answer that question, I isolated RNA transcripts from a wild type in ZR mutant grown in rich medium. And under these conditions, I expect that ZR targets will be repressed in the wild type and constitutively transcribed in the mutant. In the RNA-seq analysis, we are looking for transcripts that are just upregulated in this delta ZR background. So not only did the screen identify known ZR targets, but it revealed a very striking cluster of genes that are upregulated on this VSP2 island, which is shown here. The y-axis is the log 2-fold expression change of each gene in ZR relative to the wild type. So the taller the bar, the greater the upregulation. And those are shown in blue. You can see that 10 of the 23 genes are significantly upregulated. These include SHI-B from the first story, this 3-gene operon that contains our VER-A activator, and the chemoreceptor, along with a smattering of genes in between. So for an island that is usually silent, this result was pretty loud. And our strongest candidate for inclusion in the zinc starvation response is this VER-A operon, which is preceded by a ZIR binding site. To test if VER-A is ZIR regulated, I used this promoter region to build another LAC-Z transcriptional fusion, and I measured its activity in rich medium. So you can see that activity is very low in a wild type background, and it increases if we delete ZIR. If I then complement ZIR back in trans, we get repression. So with these two pieces of information, the presence of a ZIR box and this repressive effect, we can conclude that VER-A and its operon are likely novel ZIR targets. The regulation of AirB, however, was a little bit more complicated. We know that it's induced alongside the other ZIR regulated genes, but its promoter does not contain a ZIR binding site. So how is that possible? Well, the fact that we have a transcriptional activator just next door is a little bit conspicuous. So I next tested whether VER-A induces transcription of an AirB reporter. You can see that in a wild type background, AirB promoter activity is very low. But if we induce VER-A from a plasmid, we see a very strong inductive effect. So to summarize, under high zinc conditions, ZIR will repress VER-A, and then in low zinc environment, ZIR will dissociate from the promoter, and induction of VER-A will turn on expression of this chemotaxis gene. So this VSP2 island seems to be chock full of zinc starvation genes, some directly regulated by ZIR and others that are secondary targets. But moving beyond these transcriptional networks, I wanted to understand how these proteins facilitate aggregation. The key player here is the chemotaxis receptor AirB. If I bypass this whole transcriptional circuit and artificially induce AirB expression, you can see that our normally homogeneous wild type culture with AirB induction aggregates. So at this stage, it made sense to focus on chemotaxis. Now, chemotaxis enables bacteria to move directionally in response to chemical stimuli, and it helps them to locate more favorable microenvironments. So to give you a visual representation of this, I have a little helper, my 1-year-old niece, Valerie. So Valerie has these little spring-loaded cars, and we all sit in a circle and we pass them to each other. This is very exciting for Valerie, and you can see that she reorients in response to the nearest car. So as long as we keep the cars diffuse around the circle, she won't stray too far from the center of the rug. And this is how bacteria move in a relatively homogeneous environment. It's random, and they don't seem to stray too far from the center. But if you notice in the clip, when the cars start to pile up on one side, Valerie seizes the moment and will go after them. In the same way, bacteria sense chemical gradients and migrate along them with chemotaxis. And they do this by altering their tumbling frequency to get where they want to go more efficiently. For example, if a bacterium is heading in a favorable direction, like towards a nutrient source, it will swim for longer stretches of time without reorienting. But if it finds itself encountering the same or less of that attractant, it will initiate tumbling to get back on the right path. So similarly, bacteria can also alter tumbling frequency in order to move away from a repellent. So chemotaxis relies on the ability to sense these gradients, and that starts with a receptor protein, much like AirB. So chemotaxis receptors have two domains. They have one that binds to the chemical signal, and another that relays that information to these chemotaxis proteins that communicate with the flagellum and alter rotation. So I've confirmed that AirB does interact with these chemotaxis proteins, but what I really want to show you today is what AirB is sensing. So the AirB ligand binding region contains a PAS domain, and these have a very well-defined role in energy taxis, and they typically sense things like light, oxygen, or even redox stress. And this kind of makes sense. So if we think about the chemical gradients that exist in our culture tube, the only obvious one is a decreasing oxygen gradient as we move downward and away from the surface. So what happens if we take away oxygen? So here I'm showing you what the wild type and the ZIR mutant look like in a normal aerobic culture. You can see that the ZIR mutant forms a pellet at the bottom of the tube, and its aggregation score is closer to 0. But with the help of Sean Murphy in the Buckley lab, I was able to measure the same thing in anoxic culture tubes. And you can see that without oxygen, no pellet forms. And the culture grows pretty homogeneously. So what this suggests is that oxygen or an oxygen gradient is required for this migration. And based on studies of other aerotaxis receptors, it seems most likely that AirB is not sensing oxygen directly. Rather, many of these receptors instead monitor the redox state of electron acceptors like F.A.D. So on the left, I'm showing a crystal structure of the E. coli aerotaxis receptor that shows F.A.D in the ligand binding site. And the coordinating residues here are all indicated. I identified these corresponding residues in AirB, and I mutated them to see if that abolished function. So when I checked for aggregation, you can see that if we express normal AirB, we get an aggregate, but any of these mutant versions are not able to do the same thing. So what this suggests is that AirB may rely on F.A.D binding in order to function. So overall, AirB appears to mediate a negative chemotactics response to oxygen. When expressed, it causes cells to accumulate at the bottom of the culture tube. And what was interesting is when I dug through the literature, I came across an AirB homologue in azospirillum that causes cells to aggregate in the presence of oxygen. So it's nice to know that LTOR is not alone in doing this. To summarize this model that we built, in zinc-rich conditions, ZIR acts as a repressor of this VSP2 operon. In its absence, we get expression of this transcriptional activator, which induces a chemoreceptor, and that causes a chemotactic repellent response away from oxygen and causes our cells to accumulate at the bottom of the culture tube. So this was a very fun puzzle to work out. And something that I've been thinking about lately is why it might be beneficial to have an aerotaxis system that is turned on by low zinc availability. There's some evidence that zinc ions at the right concentration can offer protection against redox stress. So in E. coli, for example, if you starve cells for zinc by knocking out the ZNU importer, they're much more sensitive to hydrogen peroxide. And this is speculative, but I wonder if in low zinc environments, LTOR promotes aerotaxis in order to flee from oxygen or redox stress. To put this in a more biological context, there are data sets that support the idea that zinc starvation in VSP2 genes are induced in a mouse infection model and in response to chitin oligosaccharides, which are present in the marine environment. So perhaps zinc starvation is a cue for bacteria to migrate towards more anoxic regions of the gut or the sediment where they can better avoid redox stress or other stresses. To summarize these two stories, the VSP2 island was previously thought to be silent, but without ever intending to, I found two regions that are part of the zinc starvation response. And one thing that I want to mention is that there are other bacteria, a mycobacterium and C. metallodurans, that also possess genomic islands that are similarly silenced by zinc. Although the contents of these islands greatly differ, I think it's a noteworthy motif, this pairing of zinc starvation genes with a mobile element. Because zinc deficiency is encountered within the human host and likely within environmental biofilms, these findings are an important clue for deciphering the role of this island in the LTOR pandemic strain. So we're still looking to understand this island's overall function. From this work, we learned that it encodes a cell wall hydrolase that can substitute for other zinc-dependent enzymes, a transcriptional regulator and an aerotaxis system that promotes migration towards anaerobic microenvironments. And future work will attempt to characterize these other ZIR-regulated genes, examine their importance in vivo, and also search for cues that induce other components of this island. And I'd like to conclude by highlighting some of the broader lessons that I've learned along the way. So research is not always a straight line, and it's okay to follow the data even when it takes you to new and unexpected places. I learned to trust my curiosity and pay attention to the unusual and often subtle observations that happen in day-to-day lab work. And lastly, science does not belong in a vacuum. So I have grown so much as a scientist by collaborating with others and sharing my work to diverse audiences. And I think this is a good segue into some acknowledgements. So I did not do this work alone, and I want to again highlight the co-authors on these two works. This was all advised by Toby. For the Shy Bee project, we had our collaborators in Sweden, Laura and Felipe, our protein experts at the vet school, Mivanwe Adams in the Chappie lab, and 3 undergraduates, Shuning, Bree, and Camille. This work was also very much pushed forward by John Hellman and his lab, who welcomed me to their meetings, gave priceless feedback on metal-related projects, and offered protocols, reagents, and time. So thank you for being an excellent resource. I also had assistance from Sean in the Buckley lab and numerous other groups that offered materials to get me started with anaerobic cultures. So thank you to the department. I also had many helpful discussions with my lab mates along the way. And this work was funded in part by Cornell, the NIH, and the CIMID program, which actually funded Bree and Camille as summer students. But I have many, many more acknowledgements to express. Some science, some personal. And I think I can sum this up best by saying it takes a village. So first and foremost, I want to say thank you to my advisor, Toby. Possibly the best thing about working with Toby is that he matches your enthusiasm. So I could burst into his office with an exciting result and he would drop whatever he was doing and say, tell me more. So over the years, he's given me a lot of helpful advice and I want to share some of that with you. One thing is he helped me to become a better writer, and he did that through very small pieces of anecdotes and words of wisdom. So in the first manuscript that I ever sent to Toby, he sent it back and he told me to revise it using the who shot JR method. Now, this was a cultural reference that went way over my head. It was well before my time, but he explained to me that there was this whodunit mystery series in the 80s called Dallas. And at its peak, it had millions of viewers who were obsessed with finding out who shot JR. And his advice was this: storytell your science in a way that captures and guides the audience's curiosity. And to this day, that is something that I consciously think about in both my writing and when I give talks. In addition to that, he taught me to be a disciple of the mighty semicolon, which I hadn't learned how to use appropriately until I joined his lab. And sometimes, even to my dismay, he persistently tried to include archaic Latin phrases in my manuscripts, but it was always a fun back and forth that we had. As a whole, Toby was a very fun person to work for. So far, he's been willing to wear whatever Halloween costume we gave him. He treated our lab to many celebratory buffets at the local Indian restaurants over the years. And even at the peak of lockdown, he had lunches delivered to our house so that our lab meetings would retain some sense of normalcy. So above all else, Toby was supportive of me as a person first and a scientist second. Excuse me. And I think that's where the bar for mentorship should be set. So thank you. Being in Toby's lab, I had the opportunity to work alongside a really great group of people. So I joined this brand new lab in the spring of 2017 alongside Anna Weaver. And Anna and I relied a lot on each other to set up the lab, to get projects off the ground, to decipher Toby's handwriting. We were really in it together. So Anna, thank you for always being willing to help and for baking excellent baked goods. And sewing lots of masks during the pandemic. Can't forget that. We were joined in the lab by Jung Ho Shin, a very talented research scientist, and a year later by Trevor Cross. So Trevor is a fellow night owl, and I'm grateful for all the good conversation and levity that he brought to our bay over the years. Since then, our lab has grown a lot to include our axolotl mudkip. We had 2 visitors, Ziyi and Ying Li, over the years, and most recently, our lab has grown to include 4 new graduate students, Andy, Megan, Kelly, and Manuela. Each of them I had the chance to work with during the rotations, and you guys have already brought so much excitement to the lab, and I'm eager to see how your projects unfold in the coming years. I also want to acknowledge 3 very talented undergraduate students, each of them who were contributing authors to the work that I described today. They are already at or heading in really great places. And so Shuning Liu is currently in dental school. Camille Ledoux was recently accepted to a Ph.D. program, and our current student, Bree, is writing an honors thesis en route to submitting her own manuscript. So thank you, all 3 of you, for giving me the chance to grow as a mentor. My journey through graduate school is guided by my committee. I already talked about Toby, but I also want to thank Josh Chappie, who welcomed me into his lab to learn the world of proteins, and Alada Brito, who offered me a lot of career advice over the years. My 2 homes at Cornell were the microbiology department and the Weil Institute, and these were wonderful communities to be a part of. So thank you to all of the faculty, support staff, and students for just making these great places to work. Speaking of students, I especially want to give a shout out to FOMS in my cohort. Monique, Rachel, Peter, Elliot, and Anna. I'm glad that we were able to start this journey together, and I value the friendships that were forged through biosynthesis of macromolecules and just the ups and downs of grad school. So here are just a few snapshots of the fun that was had outside of the lab. I've truly enjoyed my time in Ithaca and the people that I got to adventure with. And last, but certainly not least, I want to thank some very important people who are in my life outside of science. I want to say thank you to my family who are all here today. Pictured here, my parents, my big sister Elise, my brother-in-law Adam, and Valerie, who you've already met, and my partner David. I love you all very much, and I could not have done this whole grad school experience without your unwavering support. So with that, with any luck, I will compose myself, and I am happy to answer your questions about the science. Thanks.
All right, time for questions. You can either put them into the chat window, which I will then read, or just unmute yourself and ask.
Hi, that was really good. I was curious if you thought at all about looking at whether zinc starvation might stimulate mobility of the island, because I think it's still a kind of a mystery, like how these things move and whether they might still be mobile.
Yeah, that's a really good question. And would be great to know. So a little bit of context I want to give is that the origin of this VSB2 island is thought to be from another marine bacterium. So I've always been curious, what environments would best encourage the, exchange of these islands. And within biofilms on these chitinous surfaces that induce zinc starvation, I think would be the perfect soup for that exchange. But to answer your question, no, I have not looked directly at how zinc would affect the mobility. But these islands do excise by themselves. There's a really great paper that came out just showing that all of these islands can still become circular intermediates. I played around with some of those primers and saw, yeah, it still excises, but I didn't look at how zinc would impact its transfer mobility. But I think that's a really interesting question with some really good kind of biological implications.
Cool.
I have a question.
Yeah, Peter.
Specifically about what you just mentioned about it excising. That's really interesting to me. Do you know when it excises, does it copy itself out or does it just hop out of the genome? And I guess, and if it cuts itself out, why has it stayed around this whole time, I guess? Yeah,
that's a good question. So the only gene that we know that's really important for just its excision is the final gene on this island. I think it's a transposase. Not quite sure. But so that gene, without it, it can't excise. So we know that that's an important part. I believe it exits as a circular intermediate and leaves a little bit of scar behind. I don't know if it replicates. There are some DNA replication related genes on this island. But as far as I know, it's just a circular intermediate. And as to why it would still be around if it's so easy for it to pop back out, I don't know. What I hope is that implies that there's some utility and some evolutionary pressure to keep this island, but who knows? So you, when growing it, you never end up with colonies that have just lost it? No. That's interesting. Pretty weird. Cool. Yeah, must be important, obviously, but yeah, cool, thanks. I see Steve Zinder has his hand up.
So yeah, I'm just trying to think, how do you think then, does this directly contribute to pathogenesis? I mean, you can sort of say, oh, it comes away from air and, zinc, but this makes this a more pathogenic strain in theory. So yeah, so no ideas about that.
Yeah, I mean, it could be increased pathogenicity, or it could just make it better at persisting in those reservoirs between infections. So those are two different ideas that we're thinking about. In terms of the first story with that Shy-B endopeptidase, all of these other ZIR-regulated cell wall genes seem to be very important for virulence in our hosts, but that doesn't seem very likely for cholera, just because it does have all of these other redundant enzymes that having Shy-B doesn't seem to confer any benefit. But that was my original thought, is the zinc starvation gene might give it a better cell wall that can better survive host stresses. But with that redundancy factor, it doesn't seem to be the case. But as far as this chemotaxis system, I would love to put some of these mutants into a mouse model to see, are they able to colonize different regions of the intestine? Are they hiding out in some of these more anoxic pockets? That's kind of one avenue, but I don't know if it'll pan out.
Yeah, I mean, in our gut, they're sort of, they're not like in a tissue, they're getting whatever we're eating kind of going by them. And actually all that water probably does wash out all the zinc, when you get the diarrhea. Okay.
I think Josh has his hand up.
Yeah, good question, Shannon. Very excellent seminar as always. Knowing what you know now, that you found these zinc regulators and the promoters and stuff in here, and knowing that you had to sort of go through that to figure out the right conditions of how to get these things activated, have you thought about looking more closely throughout there to see if there's other hints of other types of promoters or regulatory elements in those sequences upstream of the other genes that you have no idea what they're doing, as a way to maybe guide the right screens? And how applicable do you think that might be to figure out what the other genes are doing?
Yeah, I think that's a great idea, and admittedly isn't something that I've done. Because as the more work is coming out on that first VSP1 island, the pieces coming together, it's like, oh, maybe it's involved in phage defense. So having the knowledge of each of those genes gave it a much clearer purpose and context, and we're currently lacking that with VSP2. So I do think that is the next step.
Hey, Shannon, Scott here. Congratulations, beautiful work, beautiful talk. You've done great. But I was going to check. I assume that many cholera isolates have been sequenced from patients. And I was wondering, what kind of variability does one see either in these islands or in other regions of the genome?
That's a great question. So there is actually a lot of variability in the VSP2 island, and specifically that variability seems to be concentrated around these zinc starvation genes. So there have been recent isolates, for example, in the outbreak that was in Haiti, where this VSP2 island is there, but it has lost this chemotaxis system. It still has the cell wall hydrolase. So what that kind of suggests to me is that there isn't really stability in this island yet, given that it seems to only have entered into cholera in the 1960s. And so it's interesting to see how this might change with future outbreaks, to see which ones of these things are truly conferring a benefit, either in the host or in its reservoirs, as those are changing too.
Thank you. Great.
Any more questions? Otherwise, let's give another round of applause to Shannon. And this would conclude the public part of the defense. And everyone feel free to leave. And now Shannon, Josh, and Ilana and I are going to stick around and grill her some more.
Thanks, everyone.